and welcome to CBL. Welcome to this week's current affairs in the field of science and technology. So let's have a look what are we going to discuss this week, right? So the first thing which we are going to discuss is obviously the coronavirus and the new mutant. This new mutant has again posed a great threat to the development of the vaccine. So we'll have a look at what is this new mutant and what is actually the mutation of an RNA virus. Next, we'll discuss an important thing that is again an infection that has cropped up in Kerala, that is Shigella infection. A bit detail about this Shigella infection. The next thing which we'll discuss is Chandrayaan-2 mission's initial data released by the ISRO. We've already studied enough about Chandrayaan-1 and Chandrayaan-2, but the data has been released for the very first time, so we'll discuss that very news also. Next, we'll discuss the nano missiles. Now, what are nano missiles? What is nanotechnology? And how are these nano missiles being used in cancer treatment? That would be an important aspect which we'll be discussing in the nanotechnology in this particular news. Next, we'll be discussing the massive cyber attack in US using a novel set of tools. Last week also we had an insight into this very news. So today we'll discuss it in a bit more detail. Then we'll have an important fact on the chart, which is ICER's scientists identify the gene that greens the plants. All of us know the plants are green because of chlorophyll. So, an important discovery of the gene that helps to keep these green plants green. We'll discuss very important fact in this news. So, moving ahead, let me take on the first concept, that is coronavirus N501Y mutation in SARS-CoV-2 causes increased infectivity in mice, as the researchers said. Last week's food for thought was that this new mutation in the coronavirus has made the virus 70% more easily transmittable. So what is this mutation? A mutation means a change. Any change in the genetic sequence or the arrangement of the molecules. Since the coronavirus is a retrovirus, it has got the 4 important nitrogen bases which are arranged in a particular set of orders. Those nitrogen bases are A, U, T and G. Right? So, A, T, G, C. C is present in the DNA viruses or the DNA. So these are very important nitrogen bases which are purines and pyrimidines. They have a specific set of sequence. So when there is an alteration in any of these bases, it is called as a mutation. And mutation is something not new to the viruses. Since the very first time this novel virus was seen, it has undergone a number of mutations. Undergone mutations a number of times. Right? So how is this mutation important? Before discussing this, let us see how this virus infects our body. First of all, any virus gets attached to the target cell via the spike proteins, especially in this coronavirus case. With the help of these spike proteins, it attaches to the pulmonary surface of human body, right? The lungs surface, and thereby the infection starts. This virion or this virus particle gets engulfed into the cell. And once it is engulfed into the cell, its contents are released. It is encapsulated and the contents are released, thereby resulting and taking hold of the cell's biogenetic machinery to form its own copies. So once the viral contents are released, the viral RNA enters the nucleus, where it is replicated using the enzyme viral RNA polymerase that makes its own copies. Once the viral mRNA is formed, it helps in formation of viral proteins. So now this very cell in which the virus is already entered acts like a factory for the production of more virus particles. This cell keeps on living and it becomes a factory for the production of further viruses. And that's how the infection is aggravated and it causes severe damage to the pulmonary system. So mutation, as I told, is not something new to the virus. The virus has already gone mutations a number of times. But the most important thing is that this very mutation N501 is like making it much more transmittable. Sometimes the mutations might occur which may lead to the decrease in the virulence of a particular virus, that is its ability to cause a particular disease. Now, what is the major cause of major concern for the researchers? They want to design, and the vaccine development, vaccine is being inoculated into a number of people across the world. So, how does this mutation help or hinder the vaccine development? So, the response to the vaccine against any particular virus is because of its spike protein. Once a vaccine is designed, it is designed to target any changes in the spike protein, because spike protein is the part which is responsible for causing the infection. The vaccines are made so that there are a number of multiple regions are targeted while designing a vaccine in the spike protein. And mutation is a change at a single point. So, if there is a change at a single point, but the vaccine already made has taken into account a number of sites, so the vaccine would be helpful in combating the disease, in making the antibodies against that very infection. So, if there is one mutation, it does not mean that vaccine would not work. Vaccine would still be working and producing antibodies against that particular virus. Right? And this mutant strain in India would be studied in Jaipur Savai Man Singh Government Hospital, where the samples of the passengers who have returned from UK and tested positive via the RT-PCR technique would be taken to this hospital, and the complete genetic sequence, including the target spike protein parts, would be taken into consideration, and it would be noted how this change, how this mutant variety is going to help or hinder the vaccine development. As for now, as it was said, it greatly affects the small children and the central nervous system. But there is no such data available. The only thing that is said is that this virus has become much more, about 70% more transmittable. That's why all the flights to UK have been cancelled, and UK has imposed more bans in this festival season. So right, now from this very news, I'll move on to the next important news, that is another Shigella case in Kozhikode. Now, Shigella is an infection, a bacterial infection that affects the intestine, the colon region, and causes the diarrhea. Diarrhea, as the name suggests, it is something very commonly heard. But this Shigella bacteria is responsible for acute as well as severe diarrhea, even leading to death. The case came into notice when an 11-year-old boy died because of this infection. And subsequently, the other members of the family, they were also tested positive for this infection. But the government says, since this infection transmits through the contaminated food and water, and it has an orophycal way of transmission, then it is nothing very serious. The only thing that is serious is, if we do not care of this infection, this can even cause the death of the individual. So, Shigella infection was detected in Kozhikode, Kerala, and the symptoms are, they generally develop 1 to 2 days of the germ's entry. That means the incubation is 1 to 2 days. After that, there might be bloody and painful diarrhea, stomach pain, fever, nausea and vomiting, which is very much similar to the diarrhea seen commonly. Right? There are cases where infected persons are asymptomatic. So the good thing is that the Kerala government says this whole situation is under control. And obviously, to prevent this Shigella infection, certain things are to be kept into mind. As I told, this bacteria follows the orophycal pathway, so we must ensure to wash our hands if specifically preparing or eating food, or if we are dealing with a child's diaper. And while swimming in a pool or a lake, we should not swallow water. It might contain that bacteria. One can easily avoid eating junk food or the unhygienically prepared food on the streets. And the most important thing, that is, drink boiled water. So a small infection but was taken care of at the right time, so it does not pose any great threat to the human life, right? Next important thing which we are going to discuss is Chandrayaan-2 mission's initial data released, as ISRO said. So Chandrayaan-2 and Chandrayaan-1 mission we have already discussed in our lectures. We'll have a small insight into this. So Chandrayaan-2 was launched on July 22, 2019 and it entered into the lunar orbit September 2, 2019 to carry out a number of 8 experiments. So ISRO has said these experiments have been conducted beautifully and successfully and the data has been released. But before making the data public, it has to be transformed into a planetary data system 4 format. So it has been done, the data has been released. So a good news, Chandrayaan-2, the first data has been released successfully. All the experiments it was going to perform, the 8 experiments have been successfully performed and the data is available for the public. Right, now I move on to the next important news, that is nano missiles using nanoparticles for cancer treatment. Before understanding the nano missiles, let me tell you what is nanotechnology or nanotech as the name says. The term was given by Professor Norio Taniguchi. And the concept is very old. It came from a very eminent physicist who said, Richard Feynman, who said there's a plenty of room at the bottom. That means if we go on from a bigger level to a nano level, that is to a level that is much more smaller than the particles. So 1 nanometer is approximately 10 raised to power minus 10 meters, right? So if we go on to such a small level, the particles start behaving in an altogether different manner. For example, if I talk about gold particles, they appear transparent, and the carbon particles, they start behaving as good conductors. And other metals like silver, etc., they can be potently used to transmit the drugs. So nano world has a grade of importance in the medical field, industrial field and a number of other fields including agriculture, etc. But our today's concern would be regarding the use of nanotechnology in the formation of nano missiles. So what are nano missiles? These are actually globular particles with the hydrophilic outer shell and a hydrophobic interior. Let me show you. For example, I take this as a globular structure. So the nanoparticles are attached with their hydrophilic head outwards and the hydrophobic interiors in such a manner, which forms a globular structure, and this globular structure is called as a nanomissile. And these nanomissiles are acting as very good drug delivery systems because they can easily escape the healthy cells of the body and they can easily target the tumor cells of the body, which is a main concern while we are treating a cancer patient. We do not want that the normal physiology or the normal cells should be affected. The only effect should be on the cancerous cells. So if I talk about the cancer of the breast, lung and colon, a very important drug has been into a use, which is docetaxel. But this docetaxel was conjugated with the chemical mixture of polysorbate 80 and alcohol. And as a result, this hydrophobic drug would damage the other cells of the body such as blood cells, liver cells and lungs. So it aggravated the toxic effects. So there was a need to develop such a system for the drugs which are hydrophobic. So when these drugs are made into a nanomissile, they can easily overcome the side effects which the initial mixture had. The cancerous cells, the solid cancerous cells, have specific leakages in the blood system, blood vessels, which are not present in the normal cells. So these important drug delivery systems in the form of nanomissiles, taking an example of docetaxel, they escape the circulation and enter the solid tumors where the blood vessels are found to be leaky. These blood vessels are not leaky in the normal cells but only in the tumor cells. So there is no chance that they'll anyways affect or increase the toxicity in the normal cells. Once they enter the cancer cells, the enzymes cleave the bond between the hydrophobic missile and the drug. So once they've reached the cancer cells, the bond is broken by the enzyme and the drug is released into the target cell, thereby killing the cancerous cells. So again, a very important news, a happy news for the cancer treatment and for the cancer patients, where they can be saved from the unnecessarily toxic effects of hydrophobic drugs. Moreover, it is a much more cost-effective and a next-generation chemotherapy available for the cancer treatment. A happy news for the cancer treatment and for the cancer patients. Right. After this, I'll move on to the next news, that is a massive cyber attack in US using a novel set of tools. Last week in the Food for Thought, I'd already given you an insight into this massive cyber attack. I hope you must have read about this massive cyber attack. So let me give you an insight into this. An important company in US by the name SolarWinds that monitors and manages the IT department of the US federal government and many other private clients through its software named Orion. So the hackers used this Orion as a window and they tried to introduce a malware in the form of an update, which was not detected by the antivirus. So as soon as the update came, almost 17,000 subscribers or the 17,000 clients of this very company SolarWinds downloaded it. So this gave a window to this Trojan horse which could not be detected by the antivirus before it was detected by FireEye, the US federal department which takes care that there is no breach in the federal government and its agencies, so that there is no leakage of the data. Before it could be detected, there was an upload from the Orion software and it led to it was considered as a major cyber attack by the Russian hackers. Right, so this was an insight into this, but this was stopped by shutting down the various computers so that the malware could not corrupt it, right? So this was a massive cyber attack in US and it is said that this was done by Russian hackers. Hope you have read about it in the last week's newspapers, right? Next, I'll move on to a very important fact. Again, the ICER scientists from Bhopal have detected a very important gene which is responsible for the formation of regulation of the levels of protochlorophyllide. All of us know that the green plants are green because of a pigment called chlorophyll, and that chlorophyll is very important because it helps the plant for photosynthesis. This we have read in the smaller classes, almost in our lower classes. So chlorophyll level is very important. As soon as a seedling comes out of the soil, it must have the right amount of chlorophyll so as to ensure its survival. And this formation of chlorophyll in a plant is a series of reactions where the precursor of chlorophyll is protochlorophyllide, and this Protochlorophyllide is controlled, the level is controlled by a gene which has been detected and named as gene BBX11 by Dr. Saurav Datta, Associate Professor, Department of Biological Sciences, IISER Bhopal, and the PhD research student Nikhil. The level of this this precursor protochlorophyllide is very important, because if it is more in amount, it leads to photo bleaching. And if it is less in amount, it would not be in an adequate amount to ensure the formation of chlorophyll. And how does the detection of this very gene will help? Obviously, with the help of biotechnological tools, this would help us to design better seeds and have a better crop yield, so that to cope with the changing climatic conditions and enable the seed to tolerate the harsh conditions of the weather, so that there is a proper formation of chlorophyll, thereby ensuring the survival of the plant. So an important discovery by these two scientists from IISER Bhopal, Dr. Saurav Datta and Nikhil. Right, an important fact as far as your prelim exams are concerned. So you must remember what is the gene and how is it responsible, how is it going to help us. It is going to help us by the biotechnological tools. We can use this gene and control the amount of protoferrocolide, the precursor of chlorophyll, which will help in the formation of proper amount of chlorophyll. That's all from me. Until then, you please keep on studying. I'll see you again. Thank you.